Welcome to the Deep Dive, where we take that huge stack of global research, history, science, and, well, we boil it down into the essential stuff, the surprising stuff, just for you.
And today, yeah, we are really immersing ourselves in the world of the vine. And I think what we found it might just change how you see that glass of wine in your hand.
Or even just grapes in the supermarket.
Exactly. Because we're not just talking about wine grapes. We're diving into the sheer genetic range of Vitis species. I mean, estimates are over 10,000 known grapevine varieties worldwide.
10,000. That's staggering.
It is. So if you think you know grapes because you know Pinot Noir, Chardonnay, maybe Cabernet Sauvignon, you're really only seeing like the tip of the iceberg, maybe 1% of the actual story.
And that's the paradox, isn't it? That's what jumped out from the sources. This incredible diversity exists, but at the same time, what's actually grown is super concentrated, and it's facing threats. So our deep dive today, using reports from the OIV, that's the International Organization of Vine and Wine, it's got three main goals. First, we're going to map out the surprising geography of grape growing globally. Challenge some assumptions there.
Definitely.
Second, we have to revisit the single biggest historical disaster for vines ever, the Phylloxera crisis.
A huge one, still echoes today.
And finally, we'll get into the really cutting-edge science being used right now to try and save these ancient varieties from, well, a kind of silent genetic extinction. Okay, let's unpack this, starting with just how much vineyard there is out there and what's in it.
Right. And the concentration, it's genuinely astonishing. You have those 10,000 varieties, theoretically. But our data looking at global vineyard area shows that just 13 varieties cover over a third of all the vineyards in the world.
13 out of 10,000.
13. And get this, 33 varieties cover half, fully 50%. So we're really living in an era of, massive varietal dominance.
Okay, I have to jump in here because this is where it gets really counterintuitive. Based on the data, the world's #1 most cultivated grape variety by area, by a long shot. It's not Cabernet Sauvignon. It's not Merlot. It's not anything most people tasting wine would guess. It's Kyoho, K-Y-O-H-O.
Exactly. That's the statistical curveball, isn't it? Kyoho, 365,000 hectares. But, and this is crucial, it completely changes what we mean by global viticulture. Because over 90% of it's grown in China.
Right. Not a classic wine region focus?
Not at all. It's primarily a table grape. Big purple berries, really high sugar, like 18 to 20 bricks. And it has this distinct foxy flavor, they call it. Pleasant, but not typically for fine wine.
And it's increasing, the planting.
Oh, significantly increasing. Yes, it's a major trend.
So the single biggest player on the global vineyard stage is table grape, mostly in China. That's a huge perspective shift.
It really is. Now, #2, that's Cabernet Sauvignon, 341,000 hectares. And that one is the true international variety, grown everywhere. China, France, Chile, the U.S., Australia, you name it.
And Cab Sauv is also increasing.
Also increasing, solidifying that global presence. But then you see others like Sultanina Thompson Seedless, that's actually decreasing. Merlot is kind of stable. Tempranillo, mainly Spain, that's increasing too.
So it's a dynamic picture, and we're seeing this massive redistribution alongside trends in total area. The sources noted overall world vineyard area actually dipped a bit between 2000 and 2015.
Yeah, about 3% down since 2010, mostly driven by the EU. They were doing a lot of grubbing up, restructuring. Spain, for instance, a huge historical player, cut its vineyard area by over 10% in just 10 years.
Wow. But contrast that.
China, exploding.
A massive 177% increase in area under vine since the year 2000.
Driven largely by those table grapes, Kyoho and another one, Red Globe.
Exactly. High yield, big domestic market is a different model. Okay, this brings us to a really neat concept from the research, the diversity index. Basically, how many different grape varieties does it take to make up, say, 60% of a country's total plantings?
It's a great way to measure the genetic concentration or lack thereof.
And it shows.
Well, the findings are pretty clear-cut. Some countries are highly concentrated. Australia, Germany, South Africa, they rely heavily on just a few main varieties. And China, as we said, is extreme. Kyoho and Red Globe alone cover over 60% of their area. Very low diversity index.
But on the other end?
High diversity. You find that in the traditional European heartlands, often. Places like Italy, Portugal, Greece, Hungary, they still have this incredible library of unique local varieties, hundreds of them often still planted.
And the highest index, the most diverse?
Actually, Romania came out on top in this data set. Their top 10 varieties only make up about a third of their total vineyard area. It shows they've held on to this really wide genetic base.
That kind of diversity. It's like a living heritage.
It absolutely is. And it's a heritage that was almost wiped out entirely. Which brings us to that historical threat we mentioned. We really need to talk about Phylloxera.
Right, you hear the name tossed around like pre-Phylloxera vines, but the sheer scale of it.
It was genuinely an apocalypse for viticulture. Late 19th century it hits, lasts for, nearly half a century. It reshaped everything.
So what is Phylloxera?
It's an insect. A tiny, tiny aphid-like insect. Daktulosphaira vitifoliae, native to North America. The problem was, when it hitched a ride to Europe, probably on imported American vines, the European vines, Vitis vinifera, had absolutely zero natural defense against it. None. It just tore through the vineyards, put the entire industry on the edge of collapse.
But if it's just a tiny insect-sucking sap, why was it so incredibly fatal? Couldn't they just spray it?
That's the crucial part, the mechanism of damage. Phylloxera has two forms. There's one that lives on the leaves, the galliculus form, makes little greenish galls. That's unsightly, but generally not fatal to European vines.
Okay, so the problem isn't the leaves.
No. The killer is the other form, the radicicole form. It lives on the roots.
Underground.
And it feeds on the roots, causing these swellings. They call them nodosities or tuberosities. Now, the insect feeding itself isn't usually the direct killer.
So what is?
It's what comes after. Those feeding wounds, the swellings, they become perfect entry points for soil-borne fungi and bacteria.
Secondary infections.
Exactly. The fungi get in, rot sets in, the roots can no longer absorb water or nutrients effectively, and the vine just dies, often within about 3 years for a susceptible vinifera vine. It's a systemic collapse.
And the usual fixes didn't work. Insecticides or...
They tried everything back then, pouring carbon disulfide into the soil, flooding vineyards for months, burying vines in sand. Some had limited temporary success, but nothing really stopped it. The scale was too vast.
So the solution that finally worked?
It was biological, and kind of a Hail Mary, really. Grafting. Taking the European Vitis vinifera scion, the top part that produces the grapes you want, and grafting it onto rootstock from American Vitis species.
Because the American vines were resistant.
They were. It wasn't magic, like you said earlier. It was coevolution.
Right.
These American species, Vitis, berlandieri, V. riparia, V. rupestris, are key ones. They lived alongside Phylloxera for millennia. They developed ways to tolerate it, essentially healing the feeding wounds quickly before those deadly secondary infections could take hold.
So they weren't immune necessarily, but highly resistant.
Highly resistant. Some, like Vitis rotundifolia, are basically totally resistant. A score of 20 out of 20 on the resistance scale they developed, the Revaz index. This grafting solution, it saved European viticulture. It's why most vineyards worldwide today are grafted.
Except there's a famous exception, isn't there?
Chile, yes.
Yeah.
Remarkably, Phylloxera never properly established itself there. Geographic isolation, maybe sandy soils in some areas. Whatever the reason, Chile remains this incredible pocket of, pre-Phylloxera viticulture, a unique genetic time capsule.
That whole crisis, it really underscores why genetic diversity and conservation are so critical, doesn't it?
Yeah.
Which leads us to the modern challenge. Even with resistant rootstocks, there's this issue of conserving intravarietal diversity.
Let's break that down.
Okay, so when you talk about ancient varieties like Pinot Noir or Tempranillo, they've been around for centuries, maybe millennia. And they weren't bred like modern crops. They were mostly propagated asexually, like taking cuttings.
Exactly. Vegetative propagation. You find a vine you like, you take a cutting, you plant it. It's a clone of the parent.
But over hundreds, thousands of years of doing this.
Yeah.
Tiny changes happen, right? Mutations.
Precisely. Somatic mutations. Small genetic changes that occur in the plant cells, not through sexual reproduction. So if you start with one original Pinot Noir vine over 500 years, the cuttings taken from its descendants might slowly accumulate slight differences.
So you end up with lots of slightly different versions within the same named variety.
That's intravarietal diversity. It's the subtle genetic variation accumulated within a variety over time. And that variation is incredibly valuable. It's the raw material the vine uses to adapt to slightly different soils, climates, local pests.
Okay, what's fascinating here is the way modern viticulture works, especially with nurseries providing certified vines. It kind of works against maintaining that diversity, doesn't it?
Ironically, yes. Because modern viticulture, quite understandably, prioritizes clean, virus-free, high-performing clones. You identify one superior vine, clean it up, and mass propagate that specific clone.
Which sounds good. You get healthy vines, predictable results.
In the short term, absolutely. But if everyone starts planting only a handful of these elite clones, you drastically reduce the overall genetic variability within that variety in new vineyards. Researchers call it creating a genetic lock.
A genetic lock, meaning you've limited the future options.
You've narrowed the genetic base. You might have maximized performance for today's conditions, but you've potentially sacrificed the variety's ability to adapt to tomorrow's challenges. Think new disease strains or rapid climate change. If your entire vineyard is genetically identical, it's much more vulnerable.
So even though the American rootstock protects against Phylloxera down below, we still desperately need the diversity in the Vitis vinifera part up top, the part that makes the grapes.
Absolutely, because that's what determines grape quality, flavor profile, ripening time, resistance to other things like fungal diseases, powdery mildew, downy mildew, botrytis, esca, all those issues. Plus adaptation to heat, drought. It's all in the vinifera genetics. We need that library of traits.
Which is why organizations like the OIV are putting so much effort into standardizing how we even identify and describe all these varieties and their internal variations.
Like
ampelography, right?
Ampelography, yes. And it's incredibly rigorous. They just updated the official OIV descriptor list in 2023. It's not just looking at a leaf and saying, oh, that's Chardonnay.
No. What does it involve?
It's meticulous description. Hundreds of specific traits across the entire plant. The growing shoot tip, the mature leaf shape, the shape of the sinuses in the leaf, the density of hairs on the underside.
Serious, really?
Oh, yeah. Very specific patterns. Then there's the bud shape, berry shape, color, timing of bud burst, flowering, veraison when the grapes change color, yield components, sugar levels, acidity, even wood characteristics, plus now molecular markers like DNA fingerprinting, SSR markers to confirm identity. It's detailed.
Wow. Okay, so you identify all this variation. But how do you actually use it to create better, more resilient vineyards without falling back into that trap of just picking one super clone and creating another genetic lock?
That's where the really interesting science comes in now. The OIV has developed a specific methodology for this. It's called polyclonal selection. There's a formal resolution for it, OIV VTI 564B 2019.
Polyclonal, meaning many clones.
Exactly, but not just a random mix. It's a highly scientific approach based on quantitative genetics and statistics.
Okay, quantitative genetics. Sounds complex. How does it work in practice? Do they just grab a bunch of cuttings from old vines?
It starts with very broad sampling, yes. You need to go to the heartlands where a variety has grown for a long time, find those old Durst vineyards, and you need to sample a lot of vines. The recommendation is often 100 to 400 different genotypes, maybe 50, 70 plants per distinct location, to really capture the breadth of existing diversity.
Right. Get the raw material, then what?
Then comes the hard work. You propagate these selections and plant them in replicated field trials, often across different locations or years, and you measure everything: yield, sugar, acidity, disease resistance, all those ampelographic traits.
But wait, a vine's performance isn't just its genes, right? It's the soil, the weather that year.
Yeah.
How do you separate the good genes from just a lucky spot or a good year?
That's the core challenge, and that's where the sophisticated statistics come in, particularly tools like EBOUP, Best Linear Unbiased Prediction.
EBOUP.
Think about a vine's performance. It's phenotype P. It's a result of its genetics, G, the environment, E, and crucially, the interaction between the genotype and the environment, GXE.
That GXE, that's like a clone performing great in one region but poorly in another.
Precisely. That interaction causes instability. What EBLUP and similar statistical models allow researchers to do using data from these carefully designed trials is essentially to mathematically filter out the noise, the environmental variance E, and the unstable GXE interaction effects.
So you're trying to isolate the true, stable, heritable genetic value, the G.
Exactly. You want to identify the genotypes that are consistently superior because of their genes, not just because they got lucky with the weather one year.
Okay, so you identify these genetically superior vines. Why not just pick the absolute best one and propagate that? Why polyclonal? Why a mix?
Because if you pick just one, even the statistically best one, you risk creating that genetic lock again, just maybe a slightly better one. The power of polyclonal selection is in the mix.
How does the mix help?
You select not just one, but a group of these statistically superior, genetically diverse clones, typically somewhere between 7 and 20 different ones. You blend them together to create the planting material.
And the benefit?
Twofold. First, you still get significant improvements. The data shows you can often increase yield by 10% to 40%, boost quality traits like sugar or anthocyanins by maybe up 10%. Real gains. But the main advantage is stability and resilience,
How so?
because you have multiple, slightly different, but all superior genotypes in the vineyard together. If one clone struggles a bit in a particular year due to specific weather, or is slightly more susceptible to a particular disease strain that pops up, the others in the mix compensate. It buffers the performance, it reduces that problematic GXE effect, you get more consistent results across different sites and years. It's like built-in insurance.
So polyclonal selection isn't just grabbing random old vines, which might be called a multiclonal mix. It's a targeted, statistically validated blend of proven superior, diverse clones.
Precisely. That distinction is important. It's researched, it's quantified.
If we connect this to the bigger picture, this careful scientific conservation and use of intravarietal diversity, it sounds absolutely vital for the future.
It really is. It's about giving these ancient, incredibly valuable varieties the genetic tools they need to adapt to whatever the next century throws at them. Climate change being the obvious big one right now. It's playing the long game.
Okay, so let's recap. We dove into the global vine map, found this surprising dominance, not of famous wine grapes, but of China's Kyoho table grape, highlighting huge shifts in where and what is grown.
And the contrast between highly concentrated regions and those retaining high diversity like Romania or Italy.
Then we revisited the historical trauma of Phylloxera, that tiny insect, the root damage, the fungal infections, and the ultimate solution, grafting onto resistant American rootstock, thanks to coevolution.
And the unique exception of Chile.
Right. And finally, we explored the modern challenge avoiding genetic lock by preserving intravarietal diversity, understanding it through rigorous ampelography.
And then actively recovering and deploying it using the science of polyclonal selection, combining multiple superior clones for both improvement and, crucially, resilience.
It's painstaking work, ensuring these plants, which have been with us for millennia, can hopefully stay with us for centuries more.
Facing known and unknown future threats. That resilience is key.
Okay, so here's a final thought to leave you with, pulling back to that history. We mentioned Phylloxera's spread was partly down to things like prevailing winds carrying the winged forms. Simple, almost random factors driving a global crisis. So thinking about today's focus on global genetics, climate adaptation, intricate supply chains, what unforeseen factor might drive the next great redistribution or the next major threat to the world's vineyards? Could it be shifting jet streams? New invasive species traveling through global trade in unexpected ways? Different oceanic currents impacting coastal climates? Something we haven't even thought of yet.
That's the question, isn't it?
Something to ponder, perhaps, the next time you pour a glass of, say, Cabernet Sauvignon, a grape with European heritage, likely grown on American roots, possibly in Chile or China, representing this whole complex global story.